Hello, my name is James K. Johnson. Thank you for watching this video. This is an explainer for the article published in the Journal of Neuroscience on June 12, 2019, titled Single-Cell Membrane Potential Fluctuations, Evince Network, Scale-Freeness, and Quasicriticality. I'm the lead investigator for this paper, and I'll be taking you through a high-level overview of the content intended for a broad audience and any of my colleagues interested in a quick summary. The research question is simple. Do bursts of neural activity follow some of the same statistical patterns, whether measured from a single electrode or from an extensive array of electrodes? Now, there are two single electrode recording methods of interest here. Extracellular recording, also called local field potential, which just records the ionic currents in the material between neurons. Then there's intracellular recording, where we patch onto a cell and record from its interior so that we can measure the electrical processes of the interior of the cell. We're interested in comparing the statistics of bursts of activity across these three measures. In particular, we're measuring the geometry of these bursts, their area, their duration, and what the shape of the bursts look like as duration changes. So we gather statistics and compare them across all three levels to see if any will match the microelectrode array. This is part of a broader effort in neuroscience to try to explain the origins of scale-free neural activity. You may be more familiar with scale-freeness as fractals. Fractals have been used as art, basically anywhere black lights and bright t-shirts are popular. All fractalness or scale-freeness means is that the more you zoom in or zoom out, the more things stay the same. The statistical patterns you see don't change, don't depend on the scale. So we wondered, what does this imply for real systems? Can we get information about any scale of activity by measuring any other scale? In particular, what about single neurons? Are the bursts of synaptic inputs we see themselves scale-free? Our research group is focused on investigating whether the concept of phase transitions can be used to explain the scale-free activity we see throughout the brain. Now, you're already familiar with phase transitions. This is the transition between ice and water, only instead of varying temperature to make the transition happen, we vary the strength of connections between neurons. When connections are weak, activity at one site will on average propagate to fewer sites, so that the number of active sites in the network will diminish over time. When this is the case, we call it subcritical and say that the branching ratio, branching meaning how activity branches out, branching ratio is less than 1. As you can see, neuronal avalanches, which is our name for the bursts of neuronal activity that happen, tend to be small and not to span the entire network. At the high end of connection strength, we have the analogy to the high energy water phase of matter. Here, the branching ratio is greater than 1, and the number of active sites in the network increases with each time step, typically. As you can see here, networks connected like this are prone to explosive runaway activity that activates almost the entire network. But in between the subcritical regime and the supercritical regime, there is a point where the branching ratio is exactly equal to 1. If it's even a little off, activity will die out over time or snowball over time. This point is called the critical point of the phase transition. As you can see, the activity looks like just a combination of the other two phases, but it's not. systems at the critical point of their phase transitions have some really interesting physics that can be the same no matter what the system is made of. One of the properties which emerges is scale-freeness, but there are a number of other properties related to scale-freeness which also emerge at the critical point, and this can be used as a battery of tests to tell whether or not a system is at the critical point. We like to say that we hacked into the neural network. Like eavesdropping on a phone call, we were able to intercept the messages between other neurons to the neuron we recorded from. Now, this is important because previous ways of getting large-scale information, like a collection of informations, required the use of electrode arrays, which are arranged on a grid. But neurons are not connected on a grid. They're not even arranged on a grid. Other methods of getting the same kinds of population measures include calcium imaging, which can identify the neurons not arranged on a grid, but it still misses many of the connections between neurons and the community structure, as the field of view can overlap an intersection between different functional communities. Neurons communicate by inducing propagating waves of ion fluxes in the cell body of the neuron, the site where the neuron decides whether or not to send a message downstream. And we intersect these waves, these fluctuations of ionic currents. These messages that we're intersecting, they represent the direct activity propagating through real brain networks to a single site that is of functional importance to the rest of the brain. Single neurons both receive connections from all over the brain, and they send connections to sites all over the brain. We patch directly into visually responsive cortex. We send our tiny glass tube to the edge of a cell where we make contact. We then induce suction and start to record from the interior of the cell. Single cell recordings are not new. The current clamp technique that we're using here dates back to 70 years. However, most single cell analysis ignores the details of fluctuations or focuses on events that trigger spiking. Instead, they favor coarse-grained measures of what's going on with the interior of the cell. We gather statistics on the precise geometry of these fluctuations to get information about neuronal populations. To do this, we had to wrangle with the variability of these single neuron traces. We used running percentile detrending instead of the more popular method of detrending using a running average. This let us pool data across multiple trials and compare minute details in the fluctuations. We also used extensive surrogate testing, which lets us eliminate some very competitive hypotheses about the origins of these scale-free signatures that we are seeing. We also showed through surrogate testing that this only works for whole cell current clamp. We need the contributions of excitation and inhibition as they work out together to induce a voltage change. Supporting simulations show that current clamp and microelectrode array agreement is a hallmark of criticality. We also related this observation to the way neurons integrate information. Finally, we've given electrophysiologists new ways to answer questions about neuronal populations, even using their old data. We analyze our single neuron voltage traces and identify positive fluctuations. And then we run the same battery of tests applied to find signatures of phase transitions in neuronal avalanches. One signature is a specific criterion for fluctuation size, scale-freeness. In this case, that means that size follows a power law. The probability of having a fluctuation of a certain size is proportional to that size raised to some power, raised to an exponent. The same is true of duration. It must also follow a power law. Then, for phase transitions, or the critical point of phase transitions, there's a specific prediction for the exponent values. And it's worth mentioning that we don't get quite close enough for comfort to those exponent values. However, we do come in close agreement with the exponent values measured in other experiments, such as the microelectrode array. One of the key signatures of critical points of phase transitions is that the statistics, the physics, are universal, that we get the same measures no matter what the system is. So even though we're a little off, it may simply be indicating that we are quasi-critical, or as close to being critical as we can be without actually being exactly critical. The fact that these exponents agree with the microelectrode array is very surprising, and it establishes proof of principle that single neurons have more value than previously known. We also apply some additional tests. There must be another power law relationship between the average avalanche size or a specific duration. Then we find all the avalanches of a given duration, short durations, long durations, and we average them together, all the fluctuations or synaptic avalanches, and this gives us an average shape. We then find a rescaling factor which lets us collapse the shapes onto one another. If we get good quality shape collapse like this, and that rescaling factor predicts the exponent here, then we have some of the strongest evidence for criticality. We found these signatures in neural systems already, and we have just demonstrated them at the smallest possible scale of measuring a neuronal population. We have many trials and experiments. So this lets us do a set-wise comparison where we can tell very clearly whether or not the statistics gathered from one set are in agreement. Here, our membrane potential sometimes is power law distributed, but doesn't pass our battery of tests for criticality. Occasionally, it's not power law distributed. But the trend in the scaling indices, or the exponents, when we try to fit to a power law, is the same trend that we get when we see a microelectrode array. Now, this trend requires an explanation. What's happening, or what appears to be happening, is that every time the size exponent is a little off, the duration exponent is off to compensate. In the microelectrode array data, this has a potential explanation. That is, since you're recording on a grid, you can miss a few neurons and your data is corrupted. When you compensate for that corruption, you end up having a skewed distribution of exponent values, and this trend comes up. However, we took the membrane potential as is. There's no potential for missing neurons or corruption. So we need a new explanation for what's going on with this trend. As I mentioned earlier, we did extensive surrogate testing. In addition to using the membrane potential recordings, we used local field potential, in which case we identified fluctuations, which corresponded to bursts of excitatory activity in the network, and we tried to analyze them with the same battery of tests for criticality. We find that our predictions for the scaling relation exponent are quite poor, and that we do not have any reasonable shape collapse. Next, we used a computational method to try to capture the excitatory conductance into the cell. This measure did not produce a reasonable shape collapse-like result, and it also did not give us a scaling relation. It was not predictive. Knowing that our computational method for constructing the excitatory current fails to show evidence for criticality, we were curious if whether the longer time scale of the membrane potential is what's causing the difference, or if inhibition is needed. So we convolved the excitatory current with an alpha function in an effort to imitate a membrane potential which lacks the effect of inhibition. There, we were able to get some improvement in our predictions and something closer to a shape collapse, but all of it is significantly non-critical. Lastly, we used phase-shuffled surrogates to test the possibility that the synaptic inputs we were seeing are from uncorrelated, completely random noise, and that the power laws and apparent scale-freeness are simply an artifact. This too saw some improvement, but was significantly non-critical. They are approximately scale-free in many cases. They look to the eye like they're convincing, but they don't pass stringent tests. We also conducted some complementary modeling. With our model of 10,000 neurons, we were able to capture the full system and measure neuronal avalanches throughout the entire thing. We then defined an analog to the membrane potential, which is simply the weighted synaptic inputs to a chosen neuron convolved with an alpha function. It's already known in the model we chose that there's a parameter which controls the phase transition. So when this parameter is below 1, it corresponds to the subcritical regime. When it's equal to 1, it's critical. And when it's greater than 1, it's supercritical. It's known that this parameter implies branching. We also tested a variety of other model parameters, such as the fraction of connections between neurons which exist, and the number of neurons which are inhibitory. When we test a subcritical regime, we find that we are not able to predict the values of the network scaling from our membrane potential analog. That is, the scaling index for avalanche size has a large error when comparing the VM analog to the network. The same is true when we go into the supercritical regime. It's only at the critical regime, where scale-freeness is most rigorously demonstrated, that we have agreement between the membrane potential analog and the network. We also demonstrated an interesting mathematical result. As I said, it was known that the lambda parameter implies branching. We showed also that the lambda parameter implies a critical kind of averaging, where the firing rate of each neuron is approximately equal to a noisy estimator of the firing rate of that neuron's input population. So when lambda is 1, the firing rate of each neuron is approximately equal to a stochastic estimator of the firing rate of its input population. So we showed that critical averaging implies critical branching. The reverse is not true. You can get critical branching without critical averaging. Our work has important implications for the neuroscience community. We showed that synaptic activity is scale-free when measured at the soma of a neuron. We showed that synaptic avalanches agree with neuronal avalanches. Synaptic activity has to be correctly sequenced. When measured extracellularly with the local field potential, or an extracellular electrode, we don't get the same result. This is analogous to the way that electrode arrays or calcium imaging capture an outsider's perspective of brain function. The trend between the exponents requires a new explanation because the old explanation doesn't apply to our data. Now, criticality is hard to prove. Since we can't manipulate it, push a neural system into or away from criticality, we haven't yet shown that we're able to, then we have to rely on a different kind of a strategy. Like proving the theory of evolution, we have to collect observations over time delivered to us serendipitously. Here, we've added 3 new types of observations. We showed that in our model, inference across scales is a hallmark of criticality. In our experimental data, we showed that for these long time averages of measuring the average shape of fluctuations, we do have the ability to make inferences across scales in real neural data. There's a trend between the scaling indices, which seems to suggest some kind of self-organization mechanism. Again, the duration exponent changes to compensate for error in the size exponent to maintain a specific scaling relation. We also connected criticality to the way that neurons integrate information, in our model at least. We demonstrated that in our model, we have both a critical averaging and a critical branching. I'd like to thank my lab mates for helping me with this work. This is me. I'm the lead investigator for this paper. Caleb Wright spent years mastering the technique of multiple whole cell recordings, which let us gather enough data fast enough to make this whole thing possible. He's now a postdoctoral researcher at the Department of Biomedical Engineering and Georgia Institute of Technology. Everyone else is at the Department of Physics at Washington University in St. Louis. Shaiji, who helped me write the paper and make some figures and helped extensively with the code. Ralf Wessel is my principal investigator, who's been a nurturing and liberating advisor. Thank you very much for watching my video, and I hope that you'll be following us in the future.